Welcome back, guys. In this video tutorial we will going to talk about the complement system. This is an important part of our immunology section. So as we have not talked about the basics of the immune system earlier, so first I will try to explain the concept of the complement system in such a way that you can understand it even if you don't have any knowledge of immune system or immunology. So first thing is, what do you mean by the term immunology? So as it contains logy, so it is obvious that it is a branch of science, right? So it is a branch of science that talks about the defense mechanism of our body. So now the question is, why body requires defense system? Because there are various type of foreign invaders present that have the ability to destroy our cellular components or our organs. So what those foreign invaders are? So those foreign invaders are mainly bacteria, viruses, protozoa, parasites. Okay, so they can cause harm to our body, and that is why our body also developed immune system to protect itself from those kind of foreign invaders. Okay, so if we going to divide our immune system, so we will get two type of immune system. One is innate immune system, and another one is adaptive or acquired immune system. Okay, so what is the term of innate immune system? What do you mean by the term innate immune system? So innate immune system is present by birth in our body. Okay, that means it is always present in our body. And adaptive immune system is the system that we have acquired. Okay, so whenever a foreign invader enters inside our body, first our innate immune system fight against that foreign invader. Okay, and then adaptive immune response comes into the picture. Okay, now if we want to divide adaptive immune response further, then we will get two type of immune response. One is cell mediated immune response, another one is humoral immune response. Okay, so I think you have probably heard about the term antibody. So antibody belongs to this humoral immune response, and I think you can also heard about the term T cell, right? So those T cells are belong to the cell mediated immune response. I will going to talk about each of them in separate video lecture series, so please don't worry about this. So our today's lecture series is all about the complement system. So complement system is a part of both innate immune response and also the humoral immune response. Okay, so as its name suggests, it can complement the function of the antibody. Okay, now the question is, what is antibody? So antibody is a tetrapeptide which is usually a Y-shaped molecule. Please know about antibody for now. So this is a Y-shaped structure that we are calling as antibody. So this antibody can work against antigen. Okay, now the question is, what do you mean by the term antigen? So antigen is nothing but the immunological term of the foreign invaders. Okay, so antibody will work against the antigen. Okay, and the function of the antibody can be complemented by the complement system. Okay, that means in simpler words, the complement system will help antibody to destroy this antigen. That is the idea. Okay, so if we try to understand about the immune system in a very basic way, so immune systems is nothing but our police force, okay, that protects our city from bad peoples. Okay, so here the city is our body and polices are our immune cells. Okay, and the foreign invaders, bacteria, viruses are bad peoples. Okay, so complement will going to help the antibody to destroy antigen. Just please know about this for now. So the term complement was first given by Paul Ehrlich. Okay, and now the question is, from where the complement system is synthesized? So the main part from where it is synthesized is the liver cells. Okay, from liver cells it is synthesized mainly, although there are various other sources present. For example, your tissue macrophages, your epithelial cells of the gastrointestinal tract, all have the ability, for example monocytes, okay, all have the ability to produce complement. Okay, now those complement proteins will enters the circulation. Okay, that means complement proteins that together forms the complement system are present in the normal blood circulation. Okay, now the question is whether they are activated all the time or not. So the answer is they are usually inactivated. Okay, they are proenzyme or inactive enzyme. Okay, now when, so here I want to mention another important thing, that is, suppose, now I want to mention another thing, that antigens are present on the surface of the foreign invaders also. Okay, so let's say these two red dots are antigen. So sorry for that. So these two, okay, so now let's say these two red dots that is present on the surface of the green circle is antigens. Okay, sometimes the antigens can also present on the surface of the cell. Okay, and that cell may be a foreign cell also. Okay, and now antibody can interact with the antigen via its FAB portion. We will going to talk about what is FAB in details when we will going to talk about the chapter antibody, and this part is known as FC. So FAB means fragment of antigen binding and FC means fragment crystallizable. We will going to talk about. So via the FAB region of antibody they will interact with the antigen. Okay, under such circumstances the complement system, the complement proteins of the complement system can interact with the FC part of the antibody. Okay, and if it occurs, then the complement system have the ability to lyse the cell. Okay, so that is the normal thing how complement system works. Okay, and that is why we have told that complement system will going to augment the function of the antibody. Okay, and it is obvious the main goal of antibody is to destroy the cell or destroy the antigen, right? So complement helping antibody by lysing the cell. Okay, so as I have told you that it is present in the normal serum, normal blood serum in inactive form. So it is a globulin protein. Okay, it is a globulin protein and it is present in the blood circulation or serum in inactive form. Now the question is, if they are in inactive form, how they will be activated? So the first thing, so here I have already told you that whenever antibody react with antigens via their FAB portion, the complement will bind with the FC portion of the antibody and lyse the cell, right? That is the normal thing that they do. So here the idea is, when antibody bind with the antigen via their FAB portion, there will be a conformational change in the FC portion of the antibody. Okay, and that facilitates the activation of the that ultimately facilitates the activation of the complement proteins. Okay, so up to that part we have learned that complement systems are made up of various complement proteins, and these complement proteins are present in the normal serums. They are globulin proteins. Okay, and they usually present in inactive form. Okay, and whenever antibody binds with the antigen via their FAB portion, there is a conformational changes in the FC portion of the antibodies. Okay, that facilitates the activation of the complement. Okay, so we have already discussed that the term was given by Paul Ehrlich. So the complement system is made up of 30 serum and cell surface protein glycoprotein. Okay, so there are near about 30 proteins present that together forms the complement system. Okay, so I have already just discussed that it is an important effector arm for both innate and antibody mediated acquired immune response. Now, how complement system helps our immune system to fight against the foreign invaders? So I have already told about the lysis of the cell, right? We will going to talk about how the lysis of the cell occurs in the classical pathway of the complement. So please have patience, and you will get the opportunity to learn everything about the complement, okay, one by one. So now opsonization. Now the fact is, what do you mean by the opsonization? So opsonization is nothing but enhancing the phagocytosis of the antigen. Now the question is, what is phagocytosis? So for now, just remember that there are various immune cell presence in our body. For example, let's say macrophages that can eat the foreign invaders. Okay, and promoting the eating of the antigen by those kind of cell, for example macrophage, is known as opsonization. Okay, it also do viral neutralization. Okay, that means inactivation of the virus. Okay, it can also create some inflammatory response. We will going to talk about the term anaphylatoxin later, where you can understand how complement system creates some inflammatory responses. Okay, then solubilization and the clearance of the immune complex from the circulation and deposit them in the spleen and the liver. Okay, so now what is the normal thing? The normal thing is, this is the antigen, right? And then antibody will react with the antigen via the FAB portion. Okay, and most of the cases causes destruction of the antigen by various kinds of way. Okay, so now after once the process is completed, it is important to remove the complex. Okay, and that can be done by those complement. Okay, so it can also work as a sweeper, right? Now, so that is the basic introduction of the complement system. So now let's talk about the syllabus. So first they are telling to prepare about the components of the complement system, then activation pathways, classical pathway. Classical pathway, remember, is solely the antibody mediated pathway, then alternative pathway, then lectin pathway that includes carbohydrate, and biological consequence of the complement activation, that is your anaphylatoxin. We will going to talk about this. And last one that I will going to talk about with you extra, that is the regulation of the complement system, we will going to talk about. Okay, so now complement component include a group of proteins or glycoproteins. These components, now the question is how we can designate them. There are various type of protein. We have just discussed that there are near about 30 proteins present. So we should have some designation system. So we can designate the complement component via numerals. For example, C1, C2, C3, C4, C5, C6, C7, C8, C9. Then letter symbols, for example, factor D, and trivial names, homologous restriction factor. So you will going to understand about the C1 to C9 factor D when we will going to talk about the activation pathway. So please follow the lectures. Now designation of those C1, C2, C9 is solely on the order of their discoveries. Okay, that means C1 was first discovered, that is why it gets the name C1. C2 was second discovered, C3 was then third, C4 at fourth, C5 at 5. That is how they are not designated depending on their position in the activation sequence. Mind it very carefully. Okay, so now, so it is, I have already talked with you that complement component are synthesized mainly by liver hepatocyte. Along with that, monocyte tissue macrophages, epithelial cell of the gastrointestinal tract also produces significant amount of the complement component. Okay, another important thing that I want to mention here, that especially the C1 of the complement system is synthesized from the JD mucosa. Okay, so now in the circulation, the complement proteins are present in near about 3 milligram per ml. Okay, and the some complement component are found at high concentration. Okay, for example, C3. The C3 is a complement protein which is present about 1 milligram per milliliter, while others such as factor D and C2 are found only in trace element. Okay, so this is a first discussion on the complement system. So today we have discussed about the basic introduction of the complement system. Then we have talked about from where the complement systems are originated. Then we have talked about how we can designate the complement proteins. Okay, and then we have also talked about the amount of the complement system or complement proteins that are present in the normal serum. Okay, so from the next video lecture we will going to talk about the activation pathway, that is the main part of this chapter. So in the next video we will going to talk about the classical pathway of complement system. Till then, bye bye.